Hello everyone, I am Elias. I am a grad student with Dr. Heather Kelly, and I'm here today to talk a little bit about my project. So my project is based on identifying disease using drones and cameras to better answer questions that we have on when to do a fungicide spray, also where and what we spray. So basically we've been working with new technologies and looking for new things that are out there that can improve our disease management. So that's why we've got into drones and cameras that work together on better understanding disease in a plant. And basically a brief story about that, it is that all the light that came from the sun, it reaches the leaf surface and then it reflects back. And some of the light that we can see actually is absorbed. So what we see right here is like in this green leaf is the green, but there are so much more that we can see that is out there. And that's why we use cameras. So they help us out to see things that we cannot see by naked eye. And the same thing happens when a plant gets sick. When the disease infects the plant, actually we have symptoms before those lesions. As when we get sick, we get a fever, our plant also get a fever when it gets sick. So that's why we've been working with a thermal camera. So our thermal camera will be able to, we hope that the thermal camera will be able to identify our disease in early symptoms even before we can see with a naked eye. So we will be able to pick it up very early symptoms on our plants, and then we will know when our disease is starting and in what places of the field our disease is starting as well. But to go over our question, to justify a fungicide spray, we need to know how much our disease can actually expand, how much our epidemic goes on and how bad that can be. So that's why we've been also looking into the amount of spores that actually our plant pathogen is producing. So as you all know, our disease, like for frog leaf spot, in our spots that we have in the back of the leaf, we will have spores, and those spores will go in the air and then infect new plants. So that's why we have these spore traps right here. So they capture the spores that are in the air, and as we have right here, so we will analyze everything that we can grab from everything that we can grab from this slide coded, and then we quantify the amount of spores that are present in them. Also, we have different designs as spore traps, as the one that is on my right turn right here, which is a windsock, and our spores will get attached right here, and then we will also analyze them and compare the two different designs so far. So having that data, we'll be able to know when our disease infects and what locations our disease infect, as well as we'll be able to understand how much our disease will increase on the time. Using that information, we will be able to better trigger a fungicide spray. Besides that, we've been doing some studies on fungicide resistance to better understand what fungicide is better. And so far, as we all know, we have very well documented KOI resistance, and then we use different compounds from different active ingredients from different chemical groups, and looking over DMI fungicides, they still were looking good. They're still performing well in our trials that we're performing in lab. Well, Thank you very much.